Hello, everyone, and thank you for joining us for today's webinar on the translation of microbiome research into clinical applications. I am your moderator, Breton Pyle, and we have two exciting presenters today. We have Michalis Hadjithomas presenting on crowdsourcing the advancement of microbiome research, and we have Omar Serang that will be speaking on incorporating microbiome NGS into clinical trials. If you have any questions during the webinar, make sure to submit them through either the chat or Q&A icons that will be in your Zoom window. We'll be answering questions related to Michalis' presentation after he's done speaking. And then after those questions, we'll move on to Omar's and we'll answer any related question to Omar's presentation at the end. Now, without further ado, hand it over to Michalis to kick it off.
Thank you, Brenton. So one of the many requirements that are needed for the translation of microbiome research into the clinic is the ability to accurately characterize microbiomes using next-generation sequencing and, of course, bioinformatics. There's a huge diversity in these methods, but also there's considerable improvements that are needed. So for this reason, we have created Mosaic, which is a pre-competitive cloud platform that aims to enable the microbiome research community to accelerate the improvement of these methods through unbiased benchmarking provided in the form of community challenges. Now, most of you in this webinar are DNAnexus platform users, so you will be able to access Mosaic using your DNAnexus platform credentials at mosaicbiome.com. If you're not a DNAnexus user, you can sign up for a free account at the same website, and the only thing we ask for is your name and your email address. So again, since many of you are already users of the DNAnexus platform, I just wanted to highlight some differences that Mosaic has compared to the DNAnexus platform. Mosaic was built on the DNAnexus platform, so you will see there are many similarities. For example, we have a similar project structure, and also you can build tools in and launch them on the cloud. The major difference, however, is that the tool building experience on Mosaic is a little bit different. It's based on the WebUI. And this facilitates the ability to create tools, but also to copy other tools and improve them. So for example, what I'm showing right here is a snapshot of the WebUI that we have for tool development. You'll be able to use your browser to define the input-output specification, define the virtual machine environment, write the script that you want the app to run. And also one other difference that we have on Mosaic is the ability to attach licenses to your tools so that anybody who would like to run them will have to accept these licensing terms. We already have a bunch of tools on Mosaic that you'll be able to fork or just run if you're interested in looking at their performance. So please visit Mosaic to look at these tools. Now, as I mentioned before, one of the main reasons of developing Mosaic is to advance microbiome research using community challenges. And the reason that this is needed is because current microbiome methods are not refined enough to enable the delivery of biopharmaceutical and diagnostic products. So it's very well known that advancements are needed in many areas, scalability of the tools, their precision, their accuracy, but also their usability. So we hope that with the community challenges, we can engage the scientific community and enable the unbiased benchmarking of tools while promoting innovation. We have already launched 3 challenges on Mosaic. The first 2 were focused on the strains and the analysis of microbiome samples at the strains level. And they were both sponsored by the Janssen Human Microbiome Institute. Both of these challenges are finished. However, the third challenge, Standards, is ongoing. And for this challenge, we have the collaboration of many entities, Janssen Human Microbiome Institute, the Bio Collective and DNA Genotech, but also we're pleased to have NIST and Scott Jackson participate in this challenge. So for the webinar today, I'll go very quickly over the first 2 challenges and then discuss a little bit in more detail what we're doing with Standards and hope to get you to participate. So the first challenge was called Strains 1 and focused on evaluating the performance of different tools in profiling and assembling microbiome data. That challenge was very unique in that it provided, for the first time in a community challenge, a real biological sample for which the truth is known. And this was achieved by a collaboration between Janssen Human Microbiome Institute and Jeremiah Faith and Mount Sinai. In addition to the real biological samples, we also provided 3 simulated datasets in a similar fashion that other challenges have done in the past. Another differentiator for our challenge is that participation is immediate and also interactive. Besides the challenge datasets, we also provided training datasets for which analysis could be done and be evaluated on demand, so you could evaluate and improve your approach. Of course, benchmarking in this case is unbiased, meaning that the truth is hidden until the end of the challenge. So this challenge ended a couple of months ago, and these are the results. Cosmos ID performed the best in the profiling part of the challenge, achieving a cumulative F1 score of about 74%. One thing that I think became apparent with this challenge is that tools perform differently based on the samples that you're analyzing, whether they are actual samples or they're simulated samples. And what you can see in the right graph here is the performance of the Cosmos ID submission compared to an anonymous submission that used Metaplan for the analysis of the data. So what you can see is Cosmos ID, of course, won the challenge because they had the highest total F1 score. However, if you look at the performance across the different datasets that we provided, you will see that Metaplan performed better in the simulated datasets across all complexities. However, the Cosmos ID approach performed much better in the analysis of the biosample. So this highlights one thing, is that benchmarking should be done on biological samples and not necessarily on simulated samples, because in the end of the day, we want to use these tools in the analysis of actual biological samples. The second part that we evaluated for this challenge is assembly, and assembly is a very difficult challenge to judge or evaluate, because it really depends on what one is trying to do, on what metric they're interested in optimizing. For our challenge, we chose to evaluate the genome fraction, the ability of tools to reconstruct the genomes within a microbiome sample or the metagenomic sample. And for that metric, the winner was Ryan Schicke, who developed the MINIA algorithm and achieved a genome fraction of 99%. Additionally, at this point, I would also like to give honorable mention to a couple of other participants. Sergey Nerk, who developed Metaspace Assembler, had the longest assemblies, and also Peter McCaffrey had the best compromise between genome fraction and config size. If you want to view these results, please visit Mosaic, go to challenges, and then visit Strains 1. You will be able to look at the performances of the different submissions, both in profiling and assembly, and you can interactively dive into this data. And additionally, I invite you to visit the description of the challenge to see and listen to the webinar that we had with the winners of the challenge, describing how they approach this challenge. The second challenge we launched is the clinical strain detection challenge. And the goal of this challenge was to benchmark the ability of metagenomic tools to detect the presence of certain known strains in a sample. So this challenge had a goal to answer a very fundamental question in microbiome drug development. If we give you a certain, the sequence of a certain isolate genome and the metagenomic sequence of a microbiome, can you accurately tell us whether that microbe is present in that microbiome? So again, this is a very fundamental question, and you can see how it can be very important in the development of biotherapeutic products, but also in diagnostics. So again, the challenge here was constructed in a very unique way. We're providing actual biological samples to the users to evaluate, or data from actual biological samples. So again, the Janssen Human Microbiome Institute collaborated with Jeremiah Faith to isolate bacteria from human fecal samples and sequence both the bacteria, but also the microbiomes. And then these genome and metagenome sequences were provided to the participants, and we asked the participants to match each organism to the source microbiome. Of course, we tried to make it a bit more difficult for the participants by providing decoy isolates, for example, isolates that didn't come from any of the microbiome samples. but also microbiomes that had no correlation with any of the isolates that we provided to the users. This challenge ended on Friday, last Friday, and we hope to announce the results in September, so stay tuned. So the third challenge that we're running, and I think to me this is a very exciting challenge because it combines both bioinformatics methods but also experimental methods. And this is a standard challenge which aims to capture the diversity of protocols for NGS-based microbiome profiling methods. The reason we're having this challenge is because there is a need for accurate and reproducible NGS-based microbiome profiling methods. And it's very well known in the community that there's a variability that comes in the interpretation of this sequencing data that is due to the different protocols in sample collection and storage, for example, nucleotide extraction, sequencing technologies used, and bioinformatic methods applied. So the ultimate goal of this challenge is to capture the extent of diversity and variability across the protocols that are used around the world by different labs, and we're trying to do this by using actual biological samples. Now, ultimately, it will be a great achievement if we could also associate this, the effects of these protocol decisions with the actual interpretation of the results. But as I mentioned, the first stage of this challenge is just to collect the different methods that people are using around the world. So this is how the standard challenge is built. I'll dive into each of these steps a little bit in more detail. However, what we have done is create biological samples and DNA mixtures that are shipped for free to anybody who wants to participate. We accept the participants to sequence the samples and then upload them back to Mosaic, at which point we will perform analysis of the results and present them to the community in real time. So I'll spend a couple of minutes describing how these samples were created. We're again excited to have NIST participate in this process as well. The BioCollective and DNA Genotech have collaborated to collect stool samples from 5 different people, and then extensively homogenize the samples into master mixes, and then alicot these samples into a smaller fraction. Additionally, Scott Jackson has provided us with 2 alicots or 2 samples at varied stoichiometries that each contain the same number of isolate bacteria, and in this case, we're only using the DNA and not the actual bacteria. So we have enough material to create 700 kits that contain these 7 aliquots. There's no pre-approval needed to order these samples. You can order up to 3 kits per user. And additionally, these samples are shipped free globally. That means anywhere in the world, as long as it's legal to ship there. The only thing we ask from the participants is to commit to sequence and upload the data back to Mosaic. Once the users or participants sequence the data, they can upload both the sequencing files and the metadata to Mosaic, at which point we will automatically launch our analysis pipelines to analyze the data. And the end result of this is to provide our findings in a visual way that the community can use to investigate. So in the end, what we hope to have is 2 areas that you can use to explore the data. One is the summaries for the metadata, so you can see what other people are using in their protocols, what is their favorite, for example, library construction method, what kits they use to extract DNA, and so on. Additionally, we hope to be able to present the comparisons of the analysis of this sequencing data, and we will provide, for example, PICO-A plots so that the participants of the community will be able to hopefully see what or how the different protocol decisions affect the interpretation of the results. So you should participate in the standards effort because by participating, you will help the community. But also, you'll be able to see how your protocols and analysis of your data compare to the rest of the submissions. Optionally, if you would like, we can acknowledge you or your participation on Mosaic, but also if you're willing to participate in preparing manuscript for publication, you'll also be included as an author. One thing I should note here, this is Mosaic is a pre-competitive space, and this challenge is a community challenge. So all the data and metadata will be made freely available for download at different predefined milestones during the Standards Challenge. At this point, we are at the stage where we're shipping out samples, and if you would like to participate, you can visit Mosaic, go to Challenges and then Standards, and you'll be able to join the challenge and request samples to be delivered for you for free anywhere in the world as long as legal. So with that, I will end my presentation. I would like to encourage you to join us. You can join us in different ways. You can participate, of course, in the challenges. You can share your ideas at info.mosaicbiome.com. But also, if you want to sponsor a challenge of your own, if there's a certain part or a certain problem that you would like to have addressed, we can collaborate and deliver challenges to improve methods. So with that, I'll hand it back to Brenton.
All right, thank you so much, Michalis. If anybody has any questions, you can submit them through the Q&A and chat icons in your Zoom screen. And we're going to take a few moments here before we hand it off to Omar to answer a few questions that you asked during Michalis' presentation. So the first one right here is, what were the differences in the sample composition between the real and artificial microbiome samples in the first challenge?
So the main differences, well, of course, one was the data were sourced from real sequencing experiments. In terms of complexity, however, the real biological sample was of low complexity, so it had about 10 to 15 strains. So it was similar to the low complexity simulated data set.
All right, our next question comes in. Can we pair with one of the labs and submit only the bioinformatics portion, even if we don't win or get any reward?
So for the Standards Challenge, there is no, I'm assuming this is for the Standards Challenge because the other two challenges are already complete. For the Standards Challenge, there is no winner. I think the community will be the winner. Of course, if you want to collaborate with another lab, we encourage that. We can also facilitate this. So feel free to email us at info@mosaicbiome.com if you're looking to collaborate with a different lab. Additionally, I haven't mentioned it in this presentation yet, but what we hope to achieve in the second stage of the Standards Challenge is also incorporate different bioinformatic methods besides our own. So yes, please contact me if you're interested in providing bioinformatic methods for the analysis of the data. So the two parts, the sequencing and the bioinformatics, are totally independent parts, so you can participate in either.
Okay, great. I believe our next question here is also in reference to the Standards Challenge. Is there any preference in which sequencing technologies to use?
No, we don't provide any guidance. We hope to, of course, see a diversity of sequencing technologies. My guess is that most people are going to use Illumina and most people are going to do 16S sequencing. But we also hope to see a diversity of sequencing technologies used and also a lot of whole genome sequencing data.
All right, thank you. Now with that, we will move on to Omar's presentation. And if you do have any more questions for Michalis' presentation, please submit them through Q&A and chat, and we can answer them at the end of the webinar. All right, now without further ado, on to Omar's presentation on incorporating microbiome NGS into clinical trials.
Thanks very much, Brendan, and thanks everybody for joining our webinar. This particular clinical trials and NGS is not related directly to microbiome, but microbiome has elevated itself into NGS clinical trials sort of by virtue of the fact that you really do need shotgun level sequencing of the microbiome if you're doing live biotherapeutics or any other type of bugs into humans. This diagram reflects the use of DNAnexus in a clinical trial GXP compliant manner, actually GLP to be specific. We work with a major pharmaceutical company who's developing a live biotherapeutic for inflammatory bowel disease. And the use of NGS and shotgun sequencing of the gut microbiome was really required throughout the entire process. First of all, we needed to understand the dysbiosis state of the participants to do the appropriate patient selection. We have to understand the assay going into this live biotherapeutic. And, just the idea that we are creating drugs now from microbes is really quite fascinating and has a lot of regulatory and clinical development implications. A lot of microbiome science right now is very correlative, and obviously for regulatory purposes, the FDA and even people doing the development really want to understand what the causal mechanisms are underlying the observations. And that's where we really think the use of the DNAnexus clinical trial solution can really help. At the heart of this platform is really a shared clinical and research platform. And I say that because there are both contexts with respect to compliance, regulation, and control. And it's important to be able to move flexibly between the two and safely. You do not want identified data showing up in your research platform, but you absolutely want to be able to take advantage of clinical results relative to responders and non-responders and the potential association of response with genomic traits. The platform itself, we originally, when we embarked upon this 2 years ago, realized that we had all the analytic and data management and collaboration capabilities, but that we had to deploy a quality management system. That's a fairly big undertaking. It took us well over a year and a half to finish this. We were audited by the same multinational pharma company, and they supported us pretty much through the entire process. But at the end of that, we had everything we needed with respect to change control, continuous improvement, and really at the heart of it, computer system validation. We had everything we needed to do to plug into a GLP compliant environment. Secondly, we worked with metadata. In this case, that was the CTMS, the Clinical Trial Management System and Electronic Data Capture System that this pharma company was using. And we developed plugin for the DNAnexus applications so that any application can create a generic electronic case report form, and through the Rave network interface, the API, can deposit the ECRF right into the electronic data capture system. So we've plugged this in as well to the systems for management, potentially LIMS, and the systems of record for the results. And in this case, the results file is really an assessment of genomes and strains in this microbiome gut sample. The flow essentially goes with patients submitting their samples through sites anywhere in the world, and that's one of the advantages of this. You can integrate with sites as long as you have your data locality requirements adhered to. If the data needs to stay in Germany or the data needs to stay in Southeast Asia, DNAnexus can support that. But the key is you can open up a lot of different sites and then get better enrollment. Sequencing labs will be essentially slave to whatever management system is being run by the CRO or the sponsor. And the BCLs and the sample sheets are uploaded to the DNAnexus platform. This is all done under a very stringent security and compliance regime, and it's very flexible for users to set up the different sites and partners so that they can interact securely and simply. The bioinformaticians develop in essentially the development context, creating applications and workflows which are qualified and validated for use in a clinical context for clinical development. Clinicians and medical directors will process and review the quality and the results of the samples and essentially release them to the ECRF or to whoever is required to review the sample and approve it. The translational informatics researchers, they want to do correlative analysis on the results, anything that's happening with respect to the intervention and the association of the intervention with the participants' gut microbe makeup. Those things are going to be interesting in terms of understanding response and non-response, and of course to elevate the response through appropriate patient selection. All of this is run under a security and compliance framework, as I mentioned, and the user and collaboration management is baked into the system. And we'll look a little bit more about how the DNAnexus platform provides specific features for clinical reproducibility and clinical control. Sample management, especially if you're a CRO or you're a large lab processing lots of things, or a major pharma with many different development projects underway incorporating NGS, this gives a single site where you can review all of the different trials, different cohorts, different sites, specific tests, essentially slice and dice through the various NGS processing and the workflows as they relate to your various trials in action. You can review the lab QC, analytics QC, look at the resulting BAM reports, and ultimately approve and deliver and act upon any failed actions, and also to review any outlying cost consequences that are taking place in these pipelines. Pipelines costs can be quite variable, input dependent, and we do not want to have things at high volumes going off the reservation in terms of cost. This diagram reflects what we're seeing in terms of the ability to move between a research platform and a clinical platform flexibly and securely. We have pipelines that we can use for variant calling, mapping alignment, all the secondary analysis and interpretation. But on the clinical side, we're going to be using validated pipelines for biomarker calling, any type of signature calling, but we can also do de-identification pipelines that enable us to take phase 1 results back into a research context. Obviously, the trial has to be designed to do so, but this is a very important aspect of new trial designs and can help to really accelerate the success of the trial, the selection of participants, through understanding the relationship between the participants, their own germline and genomic makeup, and their gut microbe makeup, and their response to the intervention. The ability to move back and forth safely between preclinical research and the clinical development is enabled through the DNAnexus projects construct, which we'll talk about shortly. Again, this is all meant to be done without worrying about access control lists, buckets of data, who's running what instance. All that stuff is put underneath the hood so that the bioinformaticians, clinicians, medical directors, study designers can all work at the level of bioinformatics and NGS problem space rather than dealing with infrastructure or clouds or any of that stuff. The translational informatics, where we combine the genomic information with the phenotypic and real-world information to yield insight on the relationship between participants' genomic makeup and their response, and to do biomarker discovery, to look for signature calling, and even for potentially drug target discovery. All of that is enabled with the translational informatics solution, again, sitting on the same secure and compliant platform. Whereas the workflows for secondary analysis, de-identification, and other more batch-related jobs are essentially a transient worker model where worker nodes are spun up and their data is marshaled in order to perform an analysis on demand, and those resources are returned, and the cost of those resources is returned once the analysis is done. This is a different model. We needed persistent clusters in the form of Spark. We still need those workers to do processing, ETL, normalization, other types of activity, but we end up with a variant store. We leverage our DNAnexus GLNexus technology, which is used for joint calling and representation of variants for large numbers of participants. This has been tested with N greater than 100,000. And the trick here is to be able to have large numbers of variants represented without losing rare variants in the essentially the noise as you start stacking up 100,000 different participants. Obviously, this is more for population level research, but even when you have N's of hundreds or thousands as you move into a phase 3, this GLNexus technology is highly valuable for getting the signal out of the genomic data. The phenotypic data needs to be extracted and modeled into a common data model, and DNAnexus has worked with OMOP and CDM models in order to represent the data in a unified ontology. We're doing this with UK Biobank data. We've worked with very large-scale studies involving cohorts exceeding 150,000 with our customer Regeneron and their healthcare partners. But this activity is, let's say, doing it at scale is non-trivial, and getting to where we can capture a lot of the genomic data, a lot of the real-world data, combine it with EHR and public data sets to yield insight on this platform. This is the objective of the Translational Informatics Suite. We have visualization through Cohort Browsers, and this enables you to slice and dice and create new cohorts, share them, and analyze on them. And we have packaged analysis for things like GWAS and linkage disequilibrium analysis. All of these things are interfaced through SQL, Python, and R, giving a very rich suite, and of course, Jupyter Notebooks plug into this directly as well. But all of this enables the capture and leverage of the genomic data and the clinical results coming out of the clinical development to yield insight into the relationship between the genomic makeup of the participants and their gut microbes and their response to clinical intervention. This is an example of a Cohort Browser where we're looking at specific cancer subtypes by tumor stage. We can slice this via variants and other types of genomic information as well, and again, save these cohorts and perform secondary analysis on them. So how do we do this under the hood? All of the DNAnexus platform rides on top of multiple cloud service providers, and these world-class cloud service providers have excellent security and compliance certifications. But as I said before, when you're DIY on a cloud service provider, you're dealing with data buckets, virtual instances, storage volumes, access control lists. It's still a pretty primitive way to deal with things. And once you've built a hardened and secure and audited and compliant system, you have to run it. And none of that has anything to do with bioinformatics or clinical development or drug target discovery or biomarker discovery or successful completion of development. All of that is really stuff that is irrelevant to the process of scientific discovery. So we ride on top of our cloud service providers' excellent security, compliance, and quality certifications. But the DNAnexus platform provides data services, computation services, application services, and user interface and visualization, essentially interaction services, wrapped into this construct called a project. A project is really the core governance boundary on the platform. And rather than dealing with all of these infrastructure elements, you take your data, tools, applications, whether it's a smart cluster, a workflow to do secondary analysis, a visualization of microbiome strains or genome makeup, all of that is wrapped up into a project which can be readily shared through the platform's user and group management and collaboration features. All of this is exposed through the API and only the API. This is critical for security and compliance. As you can imagine, if our users were able to reach in and touch their data directly inside the cloud, that would bypass all of the controls and access and other types of features that I'll show shortly that enable DNAnexus to operate in a clinical environment. On top of the API, we developed the secondary analysis solution and the clinical trial and clinical testing solutions. The community solutions are a way for creating user-specific and scientific-specific interaction models to interact with the platform's analysis and data. The translational informatics we discussed, and we also have global data services coming online in combination with our healthcare partners. This is essentially cohorts of highly qualified, consented, sequenced data in therapeutic specific areas for use in discovery by our pharma companies. A brief word about the security layer cake. This gets into really the heart of what DNAnexus does to support clinical reproducibility. This is important both in a clinical development and a diagnostic delivery context. First and foremost, we run the operations, information security, quality, and compliance continuously. This is really our job, is to keep this platform running at a service level agreement, a service level that's commensurate with delivering diagnostic decisions to patients, with carrying out development tests in a clinical trial development, and to address all of the compliance issues facing our customers' regulatory audits. We don't just hand off certificates, we participate directly in your demonstration of control, release management. It's a very deep process. It has more to do with actual process of how we share information with our customers in a clinical context than even technology. But from a technology perspective, there's also a lot that's going on. First and foremost, we do all the computer system validation. This is to ensure that our systems are built and tested and deployed in a manner that ensures they are fit for specification. This is a pretty onerous requirement under GLP and the federal regulations, FDA regulations for clinical trials. All of that stuff is handled by us, and we provide a demonstration of that compliance to our customers facing their regulators. We obviously are dealing with monitoring and threat management continuously. Our platform has identity management and user access built in with all the requisite things like multi-factor authentication, federation with your directories, and other things like that. Complete encryption means practically that. The only time that data is unencrypted in this platform is when it's marshaled for analysis. Once the analysis is complete, the unencrypted data is made unavailable. Everything is encrypted at transit and at rest. That's not only in and out of our system, but also within the elements of our system. All that complete encryption is there. And given our FedRAMP moderate compliance, which we've just achieved, we've gotten the authority to operate from Health and Human Services under FedRAMP moderate. We had to really go through all of the encryption and bring it up to FedRAMP standards. Immutable data and versioned apps and chain of provenance are critical for clinical reproducibility and the ability to demonstrate reproducibility. Once data is uploaded into the platform, it cannot be changed. All applications are versioned and referenced by version. And this enables us to do chain of provenance tracking for any derived result. Any derived result can be tracked all the way back to its input files, the reference data used to generate the result, all of the configuration parameters for every step in the workflow, and capturing of all the standard error and standard IO files for all of the worker nodes that went into creating the derived result. I mentioned the data access control through the project construct, which is the governance boundary, enabling flexible encapsulation of data, tools, analyses, cohorts, visualizations, and sharing of those between users and groups. Metadata integration is how we affect the connection to upstream systems like LIMS and other clinical management systems, and downstream systems like electronic data capture or tertiary analysis systems. But the metadata integration is very flexible and is used widely to automate DNAnexus platform into upstream and downstream systems in very high volume situations for clinical diagnostics or high volume whole exome sequencing, as in the Regeneron Genetics Center. And of course, logging and auditing is foundational. Whoever did whatever through the API is captured and presented to our clinical users for use in their demonstration of control facing their audits. And that is it. And I will open this up for questions.
All right, thank you so much, Omar. Again, you can submit questions through the Q&A and chat icons in your Zoom window. All right, I'll go ahead with our first question here. What would you estimate is the time and cost for in-house building for security and compliance for clinical trials?
We look at it more as a how many FTEs you'd really need to do this. The most onerous part is really having to set up these systems that are ring-fenced and have full clinical, essentially computer system validation performed on them. We figure it takes pretty much 2 full-time employees to really be able to manage all of the development and control of a regulated set of systems. The challenge with NGS is you really have to do this in the cloud. You don't really want to build a persistent cluster locally just for economic reasons. And to do this again in the cloud, there are all the tools available to do it DIY, but this all requires essentially DevOps skills, which first of all are incredibly hard to find, specifically in areas like the San Francisco Bay Area or Boston, where bioinformaticians and other people in the technology side are in such high demand. But it is quite costly as well. And again, it is not directly related to your scientific solution. So 2 FTEs is probably a rough idea.
All right, thank you, Omar. Getting a couple questions in about microbiome data. So our first one is, what particular challenges would you say are associated with microbiome data in clinical trials in comparison to other types of genomic data?
They really do present some unique pharmacodynamic and pharmacokinetic issues. Unlike a traditional molecular drug where you have some very typical absorption, distribution, metabolism, and excretion parameters, it's much more challenging to see, are you getting engraftment? What's the state of the live biotherapeutic microbes that you put into the patient, and how are they interacting with the patient's existing microbe community? So all I can say is it kind of boggles my mind, the complexity of it. I would ask my colleague Michalis, who's much better versed in the science of this, to comment on that as well.
Yeah, so another issue is that the microbiome space is not as mature as the human genomic space. And additionally, any kind of analysis that you do in the microbiome space will be very highly influenced by the reference databases that you're using. And we know very well that there's a lot of strains out there that are still unknown or don't have have not been sequenced. So in terms of having a clinical trial, you have to be able to address those kind of issues on how do you identify something that is not in your reference database and what do you do with that. So yeah, there's a lot of challenges in terms of bioinformatics. I think in the next years, there's going to be a huge improvement in the amount of reference genomes that we have access to.
Yeah, it's very well put. If it's very difficult to discover if you don't know it's there. And after working, germline and somatic NGS for a while, the microbiome stuff, like I said, kind of twisted my brain, trying to figure out how this would all work. The other thing that we realized too, Michalis and I attended the Microbiome Drug Development Summit recently in Boston, and it's clear that strains level identification is not sufficient, and that you really do need shotgun level substrains identification to really start getting away from just correlative observation into causal analysis of the biological pathways that are being impacted by the microbe community.
So another thing that I would like to add to that, Omar, is that also the microbiome is dynamic. So you will have essentially, if you're doing a clinical trial, you will need to be sampling the microbiome at different stages. So this creates a large amount of data that you will need to be able not to just store, but to analyze. So it's one of the major differences with the human genomics, where you just sequence the human genome once or the exome of one person, and that is almost immutable. So yeah, so a cloud solution, I think, is probably necessary for any microbiome clinical trial.
All right. Thank you to both of you. All right, we got our next question coming in. How many of the clinically relevant prokaryotes have sequenced reference genomes available? How much overlap exists among the different taxa that are of clinical significance?
That's all you, Michalis.
Yeah, so I don't think even I can answer that. So we've seen in different presentations, for example, the Microbiome Drug Development Conference that we attended with Omar, that people claim the ability to reconstruct different genomes from microbiome at different success rates. So also, I think the question here is kind of like subjective, which are the clinically relevant prokaryotes. So I know that right now in the Joint Genome Institute's database, there's about 50 or 60,000 isolate genome sequence. So I cannot answer what is the overlap between those and the clinic of clinical significance. So I'll have to answer this question.
Okay. Then our next question here, is there FDA guidance in what bioinformatics methods to use in microbiome clinical trials?
Omar, do you want to take that? Or?
Well, they're starting to look at that. We did have a presentation at the same conference from the FDA, or was that from NIST? But it was around regulation of live biotherapeutics. I would have to say, in working with Precision FDA and seeing this coming online, that they're probably a little nervous about this, and they will probably try to take a similar tactic that they've done for germline standardization. But as I pointed out before, it's a really complex situation. I don't think they've made significant progress, nowhere near as significant as they've made in germline NGS in terms of standards with reference genomes that are produced by NIST with fully qualified, orthogonally validated calls, all that stuff. I think we're a long ways away from that. But NIST is being engaged in this through, as you saw, through some of the challenges, and also through Precision FDA. We're going to be working with NIST as well. So I expect it's going to follow the same trajectory, where through the guidance workshops the FDA hosted, they realized that you're not going to be calibrating your NGS, but what you're going to be doing is getting people to take biological samples that are validated, like NA12-878, running through their sequencing, demonstrating the ability to call the variants relative to the diagnostic or the device, and submitting that result back to the FDA. That's where I expect it'll go, but I think it's probably a ways off. What do you think, Michalis?
I agree with that, and that's what we have seen, with the clinical trial that you mentioned that we have running on DNAnexus. What the specific customer wanted is to be able to have different tools in their toolbox, because specifically or exactly there is no real FDA guidance what bioinformatic methods to use or which tools are accepted. So our customer is using a toolbox where they can go and use different tools that are open source and available right now in order to analyze the data. So they will be able to fully support their claims if it reaches the FDA. And that's, again, going back to the previous question, what are the differences between microbiome clinical trials and regular clinical trials? It's this lack of guidance or of established best protocols in the space. And that's why, frankly, Janssen and DNAnexus are interested in these challenges of Mosaic.
All right, thank you so much. I think that is all the time we have for today. So I'd like to thank everyone again for attending. Keep an eye on your inbox in the next couple of days. We'll be sending a follow-up email with the recording. So yes, thank you again, and have a great rest of your day.
Thank you, everybody.